on materials innovations for biomedical application and will be given by Professor Liu Bin. Bin is a Senior Vice Provost at NUS, so is well known for her research on organic nanomaterials. She has won many recognitions, such as being elected a foreign member of the US National Academy of Engineering, and also just last year, she was conferred the Royal Society of Chemistry Centennial Medal. Bin, please.
Thank you, Limson. Good morning, everyone. I tried very hard. I just came out of the plane after 18 hours flight. So my voice and hearing are not in the best shape. But I'm so glad that I have this opportunity to share with you, not only as a professor, but also as the ex-head of the chemical engineering, as well as the NUS alumni. So today I'm going to talk about materials innovation for biomedical applications. So my research group is very much focused on seeing everything. So you have heard the talk about Dean Ho just now. He's talking about how fantastic this AI is able to make a huge difference in the personalized medicine. But what I wanted to do is to see the whole process. For example, I'm interested in visualize the cells, see the cell structure, understand how the cells interact with each other, and identify the tumor cells, and then find a way to kill them. So being able to see them is not only dependent on the equipment that we have, but of equal importance is the molecules that we are able to visualize the biological process. So broadly speaking, for any luminescent materials, it can be classified into two different types. The first type is the aggregation consequential, which means these molecules are highly luminescent by themselves in solutions. But once they are induced into the nano aggregates, actually the fluorescence is significantly quenched. And that's exactly how the biological reagent actually has been used in the biological setting. It's just that we didn't pay attention. What we are interested in is the molecules with aggregation-induced emission. These molecules are not emissive by themselves, but once they're aggregated in the biological media, they give out very intensified fluorescence. So the tetraphenylethylene is one of the typical example of this kind of molecules. They're not emissive by themselves, but once they come together, they give intensified fluorescence. So here I show you some of the molecules that we designed over the past decades that all these molecules actually have the rodent structures. They have negligible luminescence as molecular species when they're very well dissolved in solution, but their quantum yield can be up to 100% in the aggregated state. That's how they're used in the biological setting. So they look extremely bright as sustained nanoparticles, and they are also highly emissive in the solid powder states. So the highly luminescent nanoparticles are very useful in the biological setting, and the high brightness as a solid powder is very useful for the light emitting devices, the organic electronics. So over the past 10 years, we actually fully take advantage of the AI molecules. They're not emissive as molecular species so that we can use them to design molecular probes that do not have background signal, but once upon interaction with the analyze will turn on the fluorescence to give you a signal. On the other hand, we are also very much interested in the other side of the extreme, which is the nano aggregates. We try to bring the organic molecules into the aqueous media and form the nano aggregates and pack them as tight as possible so that we take advantage of high luminescence of these nanoparticles as the labeling reagent to understand the biological processes. So let me give you one example. So for the AI light up probe, right, the AI molecules together with two recognition elements, they're not emissive, but once it interacts with the protein of interest, the interaction will cause the restriction of the molecular motion that will increase the fluorescence. And this will allow us to visualize the cells that are highly specific to the peptides or antibodies that we actually attach to the AI molecule. That allows us to differentiate cancer cells from normal cells, for example, as one of the applications. For those who have been actually monitoring the Nobel Prize this year for chemistry, it is rewarded to the click chemistry and bioorthogonal chemical reaction. So this is exactly one of the examples to show you how we take advantage of bioosmolar reaction, where we design an AI molecule that is not emissive, which attached with the amino acids that can automatically incorporate into the peptide glycan of the bacteria through the metabolic labeling, which means our chemical is able to integrate it onto the surface of the bacteria just by the natural process. And we designed this molecule to be cellular permeable so that we're able to specifically identify the bacteria that are already actually embedded into the, for example, human cells. Right, our goal actually is to be able to selectively label the bacteria inside of the human cell, as well as to find a way to kill them in situ. All right, so now let me switch the gear to the other extreme cases, which is the nanoparticle. So in this case, our goal is very simple, trying to encapsulate as many as AI molecules into the nanoparticle so that we're able to give you the nanomaterials with very narrow size distribution, but more importantly, tunable light emission, as well as the high quantum yield, so that we can use them for sensitive imaging, image-guided surgery, and image-guided therapy, etc. So we position ourselves is to develop the organic nanoparticles that are really the strong competitors of the quantum dots for biomedical applications. The key advantage that we have is this biocompatibility, because we can design organic molecules and organic nanoparticles that can be biocompatible and also even biodegradable. So the design and synthesis of the AI nanoparticle is straightforward. So we take advantage of the self-assembly of the block copolymers with the AI molecules to form the nanoparticle. And depends on how we select the polymers, we're able to obtain the nanoparticles with different surface functional groups that allow us to do further conjugation in order to get the nano. To further facilitate the mass production with high quality and precise control of the size, shape, and quantity, we also develop the automated synthesis via the microfluidic system. In this case, we need to have 3 syringe pumps. The top and the bottom are the syringe pump with the aqueous media, and the one as a standard is the organic media contains the polymer as well as the dye molecule that to be encapsulated. So by controlling the flowing rate or the mixing rate, we are able to produce nanoparticles with precisely defined size. For example, if the mixing time is about 5 milliseconds, we're able to produce particles of 20 nanometer. And if 10 milliseconds, the size about 40 nanometer. And if 15 milliseconds, the size about 90 nanometer. So with this strategy, all we need to do is press a button. So we're able to produce high quality nanoparticles that is independent of the researchers or the student that is included into the laboratory. And once we're able to produce particles in large quantity with high reproducibility, it allows us to continue with the subsequent biomedical applications. I'll give you one example. Just now, actually, Dingho talked about how AI is very effective in quantify the dosage, right, that is highly specific to patient. What we wanted to do here is complementary to what he talked about. What we care about is how we can visualize the cancer metastasis. We understand that for cancer patients, the most severe one is those actually undergoes cancer metastasis, that tumors actually goes everywhere, eventually become not curable. So we use zebrafish as our model, and we use our nanoparticles to label the patient tissue and inject it into the brain of the fish. And we monitor over the next 2 days, how does this cell actually migrate along the fish, so that we're able to measure the distance of the cells that travels, that allowed us to visualize and also at the same time to quantify the degree or the grade of cancer metastasis. So with this kind of visualization strategy, we're able to actually also look at how the drug dosage, the drug combination is able to kill the specific cancer tissue from the patient. So that's how we're complementary to Dingho's methodology by offering a tool that allows us to visualize the cancer metastasis as well as the treatment of different drug combinations. Now, after we build the platform, it's very straightforward for us to ask ourselves a question. Are we able to design some more exciting molecules that not only allows us to do the sensing imaging, but more importantly, incorporate the therapeutic function into the same molecule so that our nanoparticle platform will be upgraded from the imaging and sensing nanoparticle into a theranostic nanoparticle. So that drives us to develop the next generation of the AI dots at the emerging theranostic NUS. So during the COVID period, our lab has been shutting down for about 4 months. And that was a great moment that I decided to learn something new with my colleague, Wang Xiaonan. So we decided to integrate the modeling with the Bayesian optimization. And we selected about 90 donors and 93 acceptors and 13 bridges, which forms a platform of 7 million molecules. And through the machine learning process, after 7 cycles, we're able to screen about 5,000 meaningful molecules from this whole 7 million to be effective photosensitizers. And photosensitizers are organic molecules that is able to produce radicals that can effectively kill the cancer cells. And we synthesized about 10 out of the 5,000. That really gives us a lot of hope and a lot of promise to indicate how machine learning is able to significantly accelerate the progress of a chemist in the laboratory. So even we lost 4 months because of the COVID, we actually benefit a lot more from the AI-guided materials design and acceleration. It helps us to be a lot more successful in obtaining the molecules with the desirable properties. And more importantly, once our students are able to synthesize the molecules with desirable properties, they become a lot more confident and a lot happier because whatever they do, it becomes very meaningful in terms of the specific applications. So now let me show you one example how this kind of molecules can be used for real application. So in this case, we decorate inorganic nanoparticle with the AI dyes that we pre-screen on the surface for the purpose of producing a hybrid nanoparticles that offers imaging and also therapeutic functions. So in this case, we're actually looking into how the stimulate oxygen production is able to help enhance the immunotherapy. So in this case, we inject our nanoparticles into the primary tumor region, and we shine the light to induce the photodynamic therapy so that we're able to kill the primary tumors. And then the nanoparticle is able to capture the antigen that's released from the primary tumor and get trapped in the nearby lymph nodes. And from here, that's what we see here. We are able to apply the second dose of the dim light, which produces a very small amount of stimulated oxygen, but that will help to stimulate the immunocells in order to actually achieve the immunotherapy. So the result is showing on this page. We can use the light to control the nanoparticle performance in the primary tumor site with the purpose to monitor whether the treatment of the primary tumor is able to actually automatically kill the tumors that is at the distance. So in this case, we also add in the alpha PD-1, which is a checkpoint inhibitor that will be further help to enhance the immunotherapy. Now, if we look at the results, it's very straightforward. If mice with the tumor on both primary and secondary side without any treatment, actually in about 2 weeks time, the mice will suffer too much and then basically will die. Now, if the nanoparticles with a high dosage and low dosage treatment, it's very obvious that both tumors are much smaller as compared to that without the treatment. And then if we have the nanoparticle with a high dosage and low dosage to activate the immunotherapy, and together with the alpha PD-1 inhibitor, then we are actually able to inhibit the tumor growth at the distance while focusing on the treatment of the primary tumor cell. And the same result is also reflected in monitoring the tumor volume over 2 weeks time. So it's very obvious that we're able to design the hybrid nanoparticles that by addressing the primary tumor, it will show very effective treatment for the tumor at the distance. So that basically is one of the effective strategy to kill at least in mice to address the serious issue of the tumor metastasis. All right, so let me take the next few minutes to introduce to you the chemical engineering department that I'm so proud of. So what I want to share with you here is some of the cutting edge research programs that chemical engineering is leading. I understand for many of you, when we talk about chemical engineering, you will think about John Island, you will think about petrochemical oil refinery, you'll think about process control. Those were traditional chemical engineering, which only accounts to about 25 to 30% of the engineering education and the research we do at NUS Chemical Engineering. And we do something that are much more advanced and much more exciting. For example, even 8 years ago, we are among the first department in Singapore to work with the Cambridge on the carbon reduction in chemical technology. So we are working together with petrochemical companies, but our goal is not to refine petrochemical or to look at the petrochemical per se, but our focus actually is to look at how we can work with them to improve the energy efficiency, to help them use new technology to decarbonize all this energy sector. And that's why in 2018, we launched the NUS Flagship Green Energy Program, where we actually capture the carbon dioxide from the air and flue gas and extract the hydrogen from water through electrolysis and get the carbon dioxide and the hydrogen, and via catalysis, we turn the carbon dioxide into the green fuel. So that's very exciting bottom-up technology that will significantly revamp the currently top-down cracking process in the traditional chemical engineering. We're really looking to the future of the research in the next 20 to 30 years for chemical engineering. So we continue work on the decarbonization strategies, carbon capture utilization, as well as the hydrogen research and hydrogen economy. And as chemical engineering, we also have the biomolecular engineering portion, right? Chemical and biomolecular engineering. So for our biomedical engineering, we're actually leading the Singapore Pharmaceutical Innovation Program. We work with big companies, GSK, Pfizer, on the process engineering for the drug production, as well as drug formulation, crystallization. And we're also one of the initiators actually looking into the Nano Nash program that basically looking into how the nanomaterials is able to offer the treatment for the liver fibrosis, which is the common disease for people in Asia. At NUS Chemical Engineering, we're also looking into the waste to energy, water treatment, food to energy, which are hot emerging areas that are of intense research interest. We're also very good at membrane for water purification, for gas separation, for hydrogen enrichment, etc. And for the Singapore Energy Center, we're one of the contributors. Chemical engineering colleague actually is leading the world in producing the highest energy efficiency for the perovskite and organic photovoltaic hybrid solar cell. So we're really proud of NUS Chemical Engineering, not only for the research program that we're leading, but also for the super talented and delicate colleagues that I have. So for the students, you deserve the best university and also the best department. And I'm very happy and very proud to share with you that NUS Chemical Engineering has been constantly ranked among the world top 10, regardless the ranking that you're looking at. We're really on par with the best chemical engineering departments in the world. All right, so let me end the presentation by giving you a short summary. So today I talk about how we're able to actually accelerate the biomedical research and be able to visualize the tumor and tumor treatment through the AI molecule innovation. So from the conceptual perspective, we actually invented AI LiDAR probes, AI dots, AI photosensitizers, and AI bioosmolar probes. And I didn't have time to touch base on the entrepreneurship today. And one of the key features of our research is that we're not only able to publish high quality papers, but of equal importance, we're able to turn our concept into the products. So all these concepts have been successfully turned into commercial products that are available on the market through Luminicell. In terms of applications, we work on sensing, imaging, image-guided surgery, and image-guided therapy. So if you wanted to know more, you can read some of these review articles listed here. So I would like to thank my research team, and I want to highlight the amount of female PhDs that I have, excuse me, which is about 1/3 of the research group that I have. So I'm really proud of a lot of female PhD students in the chemical engineering department or in engineering in general. I'm also grateful to my collaborators as well as the funding agency. And I thank you very much for your attention. If I have more time, I'm happy to answer any of your questions. Thank you.